Assalamualaikum to my examiners, Dr. Noreen and Madam Rafida and my fellow friends. Today, I, Sufi Afrina, will present my final year project titled From Invasion to Infection, a review of understanding Salmonella pathogenic mechanism. I will begin with introduction. Salmonella species is a facultative anaerobe gram-negative bacterium that can cause various illnesses through consumption of contaminated food and water, which can result in life-threatening infection. The first alternative to treat Salmonella infection is by using antibiotics. However, it is proven that the overuse of antibiotics has resulted in multi-drug resistance. An in-depth analysis of the mechanism of Salmonella may contribute to a significant understanding of pathophysiology, epidemiology, and treatment strategies. Problem statement. It's been reported that almost 27 million cases of Salmonella typhimurium worldwide are recorded each year with 217,000 deaths. Common diseases such as diarrhea caused by Salmonella have raised a significant conflict in global human health. It calls for the development of novel therapeutic methodology to ensure proper management. There are 5 objectives to be focused on my article, which are to understand virulent factors possessed by Salmonella, such as addition and invasion factors that play a role in its pathogenicity, to gain insight into the molecular mechanism of pathogenicity of host cells by Salmonella, to elucidate the mechanism of Salmonella's virulent factor in evading human immune system, to review the antimicrobial resistance of mechanism of Salmonella which evade host response and allow successful addition and invasion, and lastly, to explain how virulent factors contribute to the antimicrobial resistant mechanism of Salmonella. Elaborating on Objective 1. These are the list of virulent factors responsible for addition in Salmonella. There are 7 significant factors that contributing in addition of Salmonella that started on flagella. It facilitates the movement and attachment of Salmonella to target host cells. The function of Fimbriae is to adhere to the intestinal epithelium, meanwhile Toll C is to promote efflux and controls the production of virulent genes. Next, lipopolysaccharide is responsible in controlling addition of Salmonella, and 4P activated gene N is involved in addition and invasion. There are 2 adenosine proteins stated here, which is SIRE that contributes to the attachment to epithelial cells and MISL that contributes to insertion and secretion protein that interacts with fibronectin. Biofilm-associated protein is also considered as addition factor as it produces biofilm for Salmonella. Move on to the list of virulent factors responsible for invasion in Salmonella. The first one is Salmonella pathogenesis island that function as translocator of effector protein. Type 3 secretory system is involved in host cell invasion. Meanwhile, cytotoxins is involved in induced pyrotosis for invasion. Next, we proceed to Objective 2, which is mechanism of pathogenicity by Salmonella. Salmonella is classified into 2 categories based on human disease, which are typhoidal serotypes that cause typhoid fever and non-typhoidal serotypes that cause gastroenteritis. In typhoidal serotypes, Salmonella initially colonize in the small intestine and penetrate through the microfold cells after immune invasion by via antigen. Then it can make it to the liver and spleen via lymphatics and bloodstream. For non-typhoidal Salmonella, gastroenteritis is brought by SPI T3SS. Infection that caused by NTS usually could subside at lamina propria. However, invasive NTS have evolved virulent genes to further infect. Addition and invasion are the crucial part in pathogenesis of Salmonella. There are 2 mechanisms of addition and invasion in Salmonella, which are zipper and trigger mechanism. In zipper mechanism, T3SS can appear as an injectisome, resembles a needle, and inject the membrane layer of host cells. It will facilitate the effectors to penetrate membrane and causing membrane arrangements and actin polymerization. The different action happen in trigger mechanism, where the invasion caused by interaction between RCK outer membrane protein to the host cell surface express EFGR. Then actin polymerization will occur due to phosphorylation of tyrosine protein and activation of pi3 kinase. In Objective 3, I will be explaining the mechanism of Salmonella in evading human immune system. In human immune system, it is divided into 2 types, which are innate immunity and adaptive immunity. Salmonella can evade innate immunity by breaking down the structure of tight junctions at epithelial cells by SPI1 secreted effectors. Besides that, Salmonella also encode gene locus SPI7 for Y antigen that will help in evading host immune response. Thirdly, Salmonella can be encapsulated in Salmonella-containing vacuole to become hidden from any detection mechanism. Apart from that, Salmonella be able to modify the antigen presentation capabilities and restrict the expression of major histocompatibility to avoid host adaptive immune response. In Objective 4, mechanism of antimicrobial resistance of Salmonella will be reviewed. The first AMR mechanism of Salmonella is intrinsic antibiotic resistance. Intrinsic resistance mechanism is the natural capacity to withstand the action of any toxic substance by utilizing structural and functional properties. There are 4 strategies utilized by Salmonella, which are inactivation of antibiotics, activating efflux pumps, modification of the target sites, and altering membrane permeability. The second AMR mechanism of Salmonella is acquired antibiotic resistance. Salmonella can acquire foreign genetic materials that makes them resistant to antibiotics through horizontal gene transfer, which consists of 3 methods, which are transduction, conjugation, and transformation. The last objective is about the mechanism of AMR by virulent factor of Salmonella. The first virulent factor to be explained is efflux pump Toll-C. From my readings, the most researched systems is the ACR-AB Toll-C system that confer resistance to several beta-lactams. It has the capacity to promote intracellular survival by expel harmful substances from the lysosome. Secondly, the PSLT-BT plasmid, the plasmid carrying TN21-like locus that resists to antibiotics such as streptomycin. It can reduce environmental stress tolerance because Salmonella may focus on maintaining AMR genes to survive antibiotics. Next, biofilm. Extracellular polymetric matrix in the biofilm function as a strong barrier that prevents therapeutic medication from penetrating. Last but not least, gene mutation. It will initiate resistance to specific antibiotics. For example, mutation of RPOA gene will losing bacterial fitness and causing resistance to rifampicin. In conclusion, this review contributes to the understanding of Toll-C protein as the predominant addition and invasion factors of Salmonella due to its significant role in regulating virulent genes. Salmonella also employs the Toll-C-dependent efflux mechanism for pathogenicity, which plays a crucial role not only in addition and invasion of host cells, but also supports the zipper and trigger mechanism involved in this process. Furthermore, Salmonella evades the immune system through mechanisms like modulation of the innate immune response and inhibition of antigen presentation. Next, activation of efflux pumps in intrinsic AMR mechanism of Salmonella is a crucial strategy that contributes to successful addition and invasion. Lastly, it is proven efflux pump Toll-C is a remarkable virulent factor of Salmonella that aid in mechanism of AMR by Salmonella. My recommendations for my review study are to further study about the role of Toll-C-dependent efflux mechanism in promoting addition and invasion so that potential inhibitors can be developed to block this efflux system, and to investigate further the AMR patterns of Salmonella in the community settings so that the impact on public health could be further understood and potential strategies to combat the AMR could be developed by the health authorities. That's all from me. Thank you for watching.